Hi, my name is Zach,
I'm Kenneth.
and our topic is prions.
The first question is, what are prions? Prions are misfolding proteins which are capable of converting normal proteins to a prion formation. Prions are cause of disease such as croup in goats and mad cow disease. Prions affect humans when they consume prion-infected animals or when their proteins naturally misfold. What agents convert a protein into the prion conformation in the first place? Current research provides the public ideas about how environmental agents cause the prion formation and the method to repress this process. It is known that LSB2 protein is contributed in the prion formation by transfer the sub-35 to the prion formation PSF+. Recently, scientists told us the thermal stress can increase the amount of LSB2 protein. We can see the data in the picture. After the heat shock at 39 degrees for 2 hours, we can get the result by observing the colonies on the selective medium. The result is interesting. The colony frequency of the heat shock group from 0.1% higher than the control group. Then we are moving to the LSB2-like group. There is no change about the colony frequency. Therefore, we can conclude that the thermal stress enhances the ability of LSB2 protein to transform the normal protein to prion, and none of them can work under this mechanism dependently. Normal disease can be treated with antibiotics or antiviral medicines, but not a protein. It is difficult to denature a prion because they are open chaperones, which help fold a protein into the proper conformation. Meaning every time they are degraded, the chaperones will refold it into the prion conformation. Recent research also proved that the prion formation can be controlled by the RAG disruption. The RAG is ribosome-associated complex, which is composed of HSP4001 and HSP70SSZ. The lack of each one will cause the RAG deficiencies. SSP is a chaperone in cell, which has functions to repress the prion formation. According to the researchers, the RAG disrupt group can wipe out with prion PSF+ only when there is overproduction of HSP104. We can see the result here. The colonies with the overproduction of HSP104 are almost disappeared. The mechanism of the inhibition of RAG deficiencies is it leads to the release of the SSP chaperone to the cytoplasm. More SSP were found in the top fraction by researchers' testing. That means the SSP was dissociated with the ribosome. Once the SSP was released, it would antagonize the SSA chaperone and then inhibit the propagation of prion.
Typically, prion treatment tends to focus on disrupting the complexes formed. This has two targets, the prion chaperone complex and the prion protein complex. One research group is looking to regulate the folding properties of SHA-2, a prion protein. What they are focusing on is the formation of a lipid raft, a detergent-resistant membrane that forms on the complex of SHA-2 prion and PRPC to make PRPSC, the prion isomer. The research demonstrates that any alteration of the lipid raft protecting the complex will lead to misfolding of the protein into the prion. The lipid raft of the SHA PRPC complex was controlled by the expression of a chaperone TRAP1. So the researchers were trying to identify the chaperone TRAP1 as a target for regulating the misfolding of PRPC. Here the researchers are establishing successfully repressing TRAP1 expression in the second group to help demonstrate their findings. The amount of TRAP1 revealed by immunofluorescence analysis is much lower in the RNA targeting group. When the groups were lysed and analyzed for levels of SHA, PRPC, and floatin-2, but the last one isn't important for the topic at hand, the gradients show the relationship between chaperone TRAP1 helping prevent PRPSC formation. The other way to inhibit the prion formation that I mentioned earlier was to interrupt the complex directly. A research group led by Hoover also looked into the lipid raft, but instead of the chaperones in charge of the complex, they looked at actually inhibiting the lipids so the raft couldn't form at all. The actual graphs are a bit difficult to read, but you can see the lack of a positive prion indication on the western blots for the KDA and the groups with the brain lipids to inhibit the lipid raft formation. So why should you care about this? For a start, some human inherited diseases, such as Alzheimer's and CJD, have been linked to prion activity. The current research still hasn't found a way to reverse prion damage, but it is finding ways to slow it down.
Prions are still uncharted territory for medical science right now. We don't fully understand how they work, just what they do. The best way to help yourself is to try to avoid prion infection from the beginning. Simple things such as buying food from quality food distributors and taking care to avoid high fevers to induce prion folding go a long way.
If I fall again, will it be the end? I know it's wrong, you think I'm strong, but I just pretend. It's taking over, will it bury me? Or will clarity become the cure for my disease?